Hello and welcome to this video lecture on section 40.2 on the components of the blood as we continue discussing chapter 40 on the circulatory system. Our objectives in this video are to list the basic components of the blood, compare red and white blood cells, and describe blood plasma and serum. So first we'll talk about the functions of blood. We are going to maintain our homeostasis by stabilizing pH, which, if you remember, our pH scale is from 0 to 14, 0 to 6.99 being acidic, 7 being neutral, and 7.01 to 14 being more basic. And our human blood should be really within the range of 7.35 and 7.45, 7.4 being the sweet spot. And our temperature of our body. We know that our body temperature should be at 98.6 degrees Fahrenheit, and we have to have a homeostasis for our osmotic pressure, which is the minimum pressure that we need in order to stop movement of our water across a semi-permeable membrane from a region of maybe low solute concentration, maybe an area where I would think of it as having some type of fluid that isn't as salty, move to an area of high solute concentration. So we want to maintain that, right? And by eliminating excess heat. And we could do this through mechanisms like having vasodilation in the vessels that are closer to the surface of our skin so that that heat from the blood can radiate off into our environment. We also are going to support growth by distributing nutrients and hormones by removing waste, and we can have our blood serve as a protective role against things like viruses. And we can also transport clotting factors and platelets in order for us to prevent blood loss, and we can transport our white blood cells to sites of infection. Now let's break down the components of our blood. We can see this test tube over here that has had blood centrifuged, meaning that we've spun that test tube of blood round and round and round until we start to see some of the components separate. So we can see at the bottom here, it's kind of including this clear grayish area known as a buffy coat, and those would be our formed elements of our blood, which consists of our red blood cells, our white blood cells, and our platelets right in here. The rest of our blood is made up of plasma. So let's start off by talking about hemoglobin. We're going to find hemoglobin in our red blood cells, so in the cells in this bottom portion of the test tube, and our hemoglobin helps us to distribute that oxygen in our closed circulatory system. And I say closed because we're talking about humans here, our vertebrates, and then a lot of our invertebrates as well. So that's what the job of our red blood cells down here is. It's to carry that oxygen and distribute it to our oxygen deprived tissues. We also have our white blood cells I mentioned down over here, and their job is our immune response. And we also have our platelets, which are really cell fragments. They come from a cell called a megakaryocyte, and they can help clot our blood when needed. And then back to our plasma here, which makes up 55% of our blood. This is the liquid portion of our blood, and it's going to contain mostly water. Look, 91% is made up of water. And then we have some proteins in there. We call these plasma proteins, which consist of albumins, globulins, and fibrinogens. And then we'll, of course, have other solutes. We've got some salts, lipids, and glucose, vitamins, and things like that. And if we look at where all the water is within our body, we see that 66% of it is intracellular, meaning that it is within our cells. The other 33% is extracellular. 25% can be found in our vessels as a part of blood, and then we have the other 75% of that as the fluid interstitially, meaning within our tissues. And this is a lot of what I've already kind of talked about in the previous slide, where we have the bottom component here making up our formed elements. We don't say cells because even though we have red blood cells and white blood cells, remember that our platelets, even though they're called thrombocytes and the suffix "cyte" means cell, they're really cell fragments. Now let's talk about where these cells come from. We go through a process called hematopoiesis that is going to take place in our bone marrow here. In there we've got these stem cells, and the stem cells can give rise to our white blood cells and red blood cells and our platelets. So you could see these precursor cells here. So we'll start with the erythroblast. Erythro means red. Blast is going to let us know that that is immature. I always think B for building, like we're still working on it. And then that is going to turn into a red blood cell or a mature erythrocyte. These here are representing some of our precursors to our mature white blood cells. So we have our lymphoblasts that turn into lymphocytes. Our monoblasts are going to turn into monocytes, which when these cells move into the tissues, they become macrophages. And then our myeloblasts can differentiate into 3 separate cells. And soon we're going to talk about how these are known as our granulocytes, meaning that they have granules that we can visualize when we use certain stains. So first we have our neutrophil. You could see that their granules are a little bit more neutral appearing. And then we have our eosinophil, which will show up with red granulocytes, or granules I should say. And eosin means red as well. Phil means loving actually, so loving the color red there. And then basophil, baso I think of blue, and these granules turn up blue when we use certain dyes. And lastly, we've already mentioned that the megakaryoblast is going to break off portions of its cell, and those cell fragments become the platelets and aid in blood clotting. So we're going to dive into detail first with our red blood cells. These are biconcave shaped disc cells, so you can see that over here, and they have that shape so that we can increase surface area. When we have more surface area, we are able to have more space in order for us to contain more hemoglobin. And this is going to be a pigment that binds oxygen. This is why our red blood cells are actually red. The heme portion, which if you look at this figure down over here, you could see the heme portion here is going to bind up to 4 oxygens. So you can see some of the oxygen bound here. It does have the ability to bind to carbon monoxide. It actually has a great affinity or desire to bind carbon monoxide, which is why it could be so dangerous when we do have it around us, because it will bind to our hemoglobin molecules, displacing the oxygen. When we do bind oxygen to hemoglobin, we call that oxyhemoglobin. And then when we take that oxygen off, let's say maybe to give it to oxygen-deprived tissue, then we are going to call it deoxyhemoglobin. Invertebrates can use other forms of this. You could see the molecules over here. We have our hemocyanin, which is going to be a blue-green copper-containing protein in mollusk crustaceans and some arthropods. And then we have our heme erythrin, which is a red iron-containing protein in some worms and annelids. Now let's talk about carbon dioxide's transport in the blood. Now 7% of the carbon dioxide just dissolves within the plasma, that watery portion of the blood, whereas 23% of it is going to bind to the globulin portion of the hemoglobin. Let's quickly look at this molecule of hemoglobin to see the difference. So you could see this red disc here is representing the heme that again is going to have an iron molecule in the center that will bind the oxygen. Now when we talk about carbon dioxide binding to the globin, we're referring to these areas that are like a neutral brown color. And so that is where the carbon dioxide is going to bind. but the majority of our carbon dioxide is transported in the plasma as bicarbonate ion, about 70% of it. So this is what happens. We have our carbon dioxide, it combines with the water and becomes carbonic acid. It then is going to dissociate, and we are going to have a hydrogen ion release and a bicarbonate ion. So it stays as this bicarbonate ion as it moves through the blood in its plasma, and then let's say we have that blood travel all the way over to the lungs, I should say the pulmonary capillaries, and then it can revert back to carbon dioxide in the alveolus and be removed from the body through expiration or breathing out. Now, we've talked about how our red blood cells are produced in the bone marrow. And once they're produced, we will have an immature version of it. And once they lose their nucleus and most of their organelles, again, to make more room for hemoglobin, they will be mature. Without that nucleus, though, they can't make proteins for cell repair, so they only live about 120 days. The old worn out cells will be removed from circulation through macrophages. Remember, we talked about those white blood cells called monocytes, and when they travel into the tissues, we call them macrophages. So the tissues we're referring to here are within the liver and the spleen. Now, this disc shape, like we said, is going to be important in their role in our circulatory system. It allows them to squeeze through small capillaries and allows them to have that maximum surface area for gas diffusion. And a very important hormone is going to allow us to make more red blood cells since they only last 120 days. This is erythropoietin, or EPO for short. This hormone is produced by the kidneys. We can see the kidneys here. And sometimes it is due to a response of low oxygen in our blood. So our kidney is going to secrete erythropoietin that travels to our bone marrow, and the stem cells are going to increase red blood cell production. We then get more red blood cells. Our oxygen blood levels are going to return to normal, and we no longer will have the kidney secrete erythropoietin because we have maintained homeostasis. Now, some people have engaged in blood doping where they work to increase the number of red blood cells and will sometimes do this by injecting erythropoietin. This is going to increase the amount of red blood cell production, meaning that they will have better oxygen delivery to their tissues, so it can increase their stamina and athletic performance. Although it's very dangerous because with that many cells in our blood, it can become too thick or viscous and it could cause heart failure. Some other disorders with red blood cells are jaundice, where we get an accumulation of heme in the blood if our liver can't excrete it properly. So we start to see the skin and the whites of our eyes turn yellow. You may have already heard of anemia as another disorder where we have too few red blood cells or too little hemoglobin. It could be due to an iron deficiency because, like we said, the iron needs to be a part of heme in order to bind oxygen. This is the most common form. And then if we have an adequate dietary intake, we aren't able to make hemoglobin. There's also pernicious anemia, which is a lack of vitamin B12, which is needed in order to make red blood cells. If we don't have vitamin B12, our DNA synthesis is going to be impaired, but RNA and protein synthesis can continue, which makes the cytoplasm grow without the nucleus dividing properly. And so the result will be us having large, immature, fragile red blood cells, and we call these megaloblasts. Another type is going to be folic acid deficiency anemia, where we are going to need that folic acid in order to make our red blood cells as well. Once again, if we don't have this folic acid or folate, we won't have proper DNA synthesis and the cells can't divide normally. Hemolytic anemia is when we have too much hemolysis or rupturing of our red blood cells. And then sickle cell anemia, which you may have heard of, is a genetic disease that's going to cause the red blood cells to become sickle-shaped or prone to rupture. This is due to a defective hemoglobin structure that changes the shape of the red blood cells. Now let's shift into our white blood cells, or our leukocytes. Leuko means white, cyte means cell. And these are going to be large cells and are the only formed elements that have a nucleus. They're going to be translucent unless we use a stain to be able to see them. They are less numerous than red blood cells, which is why when we go back to that test tube, it's only going to be found within this little gray layer here, and it makes up less than 1% of our blood. So we will have them produced in our red bone marrow. Their production is regulated by a colony stimulating factor, and so that is what is secreted. When we look at this graph over here, the factor stimulates these stem cells over here. Going back and forth too much. And their job is to fight infection, and they'll be an important part of our immune system. You'll see this when we discuss the immune system in class. And some are going to live only a few days, while others are going to live months or years. So we classify the white blood cells as granular leukocytes, or sometimes we just call them granulocytes, and agranular or leukocytes, or agranulocytes. So that A in front of it means without granules. The granular leukocytes, like I said, have noticeable granules when we dye them, and we'll also see that they have lobed nuclei. So that's going to consist of our neutrophil, eosinophil, and basophil. So you can see this neutrophil has, I would say, 3 to 4 lobes. We've got 2 lobes here for our eosinophil and 2 lobes for the basophil. Our agranulocytes have no granules or very little granules, and they have non-lobar nuclei. So we see a nice round nuclei here for the lymphocyte, and then almost like a kidney bean shape or cashew shape type of nucleus for our monocyte. Let's go through each of these white blood cells to know what they're all about. Our neutrophils could be pictured over here. You can see their lobed nuclei are stained in purple here, and they are the most abundant white blood cell. They make up about 50 to 70% of all of our white blood cells. And they're going to be the first responders to bacterial infections. They engulf pathogens through phagocytosis or cell eating, and they can leave the bloodstream so that they could be found in interstitial fluid and our lymph. And here you could see one of those neutrophils going through the process of phagocytosis. And they are being pictured here, showing how they're at a resting state within our blood, and then when they are being called into action, they are going to have these receptors that are going to adhere to these little knobs or treads that you can think of that are going to slow the cell down, kind of rolling along the wall of the blood vessel until it can find a little opening. Because if we're having some type of infection taking place, the vessels close to that area will become more permeable, meaning it's going to allow cells to move through. So these white blood cells will squeeze themselves through the endothelium that lines the blood vessel and move into the tissue. You can also see that taking place over here. This is our blood capillary, and our white blood cells are squeezing their way into the tissues. Next up are our eosinophils, and you could see the red granulations in the cell here, here, and here, and we see that their nucleus is bilobed, and their job is to function in parasitic infections, and they also play a role in allergies. Our basophils are pictured here with their large blue granules. They are the rarest of our white blood cells, and they have a U-shaped or lobed nucleus. In our connective tissues, our basophils and mast cells are going to work together to release histamine during allergic reactions. So eosinophils and basophils and mast cells all work together to create allergic reactions and are releasing histamine so that they can dilate the blood vessels, but it'll constrict breathing passageways, which is what is going to cause an asthma attack to take place. Our lymphocytes are found in about 25 to 35% of all of our white blood cells, and there's two different types, our B cells and our T cells. You'll also see when looking at these cells that they will have very little cytoplasm. Their nucleus takes up a majority of the space. Now let's talk about the B cells first. When they mature, they differentiate into what are called plasma cells that are going to produce antibodies, which help to mark the pathogen in our body, let's say like a bacterial cell, so that our immune cells know to go and destroy it. Our T cells, especially our cytotoxic or killer T cells, are going to be able to directly destroy pathogens. And then we have our monocytes, which are the largest of our white blood cells. In our tissues, they develop into larger macrophages, which engulf the pathogens because they can undergo phagocytosis. And they not only eat up the bad foreign pathogens, but also our old cells and debris. Now let's talk about some of the disorders involving white blood cells. There is this disorder known as severe combined immunodeficiency disease, or SID, which is an inherited disease in which lymphocytes don't develop, meaning that both the T and B lymphocytes don't work properly, and this leaves our body unable to fight infections. Even minor germs can be life-threatening. So one of the most famous cases was David Vetter, or Veter, I'm not sure how you pronounce the last name, but he was known as Bubble Boy, and he had to live in a completely sterile environment to protect him from infection. SCID can now be treated with bone marrow transplantation or gene therapy, and this offers hope for a more normal life. Then we've got leukemia, which is a group of cancers in which white blood cells proliferate without control. Most of these white blood cells are abnormal, so they're not functioning as well. And then we have infectious mononucleosis, which is also known as mono or the kissing disease. And this is caused by the Epstein-Barr virus, which infects our lymphocytes and results in someone feeling fatigue, a sore throat, and swollen lymph nodes. Now let's talk about the platelets and blood clotting. Platelets, again, are also called thrombocytes, and they are those cell fragmentations from a megakaryocyte in our red bone marrow. So you can kind of see those fragments coming off over here as well as over here. So we can't really say that they are a true cell, but we do have about 200 billion platelets that are made per day. They are going to function in blood clotting, as we said, which is also known as coagulation. Now, our plasma proteins, prothrombin and fibrinogen, are also involved in the clotting process. So we absolutely need them along with vitamin K so that we can form prothrombin. And this is just a vessel wall that's damaged, and here we are showing you the clotting process where we have fibrinogen turning into the fibrin fibers and catching all of the red blood cells to form that clot. Now we need to do that to make sure that our plasma and formed elements don't leak out of our broken vessels. There's actually 13 different clotting factors, our calcium ions and enzymes that work together to create a clot. When our vessel breaks, the platelets are going to clump to partially seal it, and we could see this pictured over here. And then the platelets and injured tissue are going to release a clotting factor known as prothrombin activator. This is then going to convert our prothrombin into thrombin, and then thrombin will convert fibrinogen, which is inactive, into the active fibrin threads, which we said will help to catch all of those red blood cells and form the clot. Now notice that these are going to require calcium ions. Now, I mentioned a lot of this already, but that fibrin clot is just temporary, and we could see that clot nicely over here. Then there's an enzyme called plasmin that will destroy that fibrin network so that tissue cells can grow to repair the vessel. Then the serum can escape from the clot, and that contains all the components of plasma except for fibrinogen and prothrombin. What are some disorders related to blood clotting? We have thrombocytopenia, meaning that we have too few platelets, and this is due to not enough being made in the bone marrow, or maybe there's increased breakdown outside of the marrow. This can be caused by leukemia or drugs, and as a result, we can have excessive bleeding. There's also thromboembolisms. When we use that word thrombus, it's referring to a stationary clot, meaning the clot is not moving and could be attached to a vessel. When we use the term embolism, that means it's a clot that is traveling through the blood, and this could be very dangerous because it could plug a vessel. It can be especially dangerous if it travels and goes to an area like the lungs, which could result in a pulmonary embolism. Then we have hemophilia, in which it is a genetic deficiency of a clotting factor and doesn't allow the body to form clots. And so once again, we can have excessive bleeding. And here's just some images showing you what that excessive bleeding can look like, although there is replacement therapy to help with people who have hemophilia. Now let's talk about plasma. It's going to consist about 91% of water and 9% salts and organic molecules. The solutes are helping for it to maintain an osmotic pressure of blood, meaning that the salts are acting as buffers, essentially making sure that we attract enough water into the blood, but not too much. Other solutes in there are nutrients, waste, and hormones, and we've already talked about the plasma proteins, which are abundant, such as albumin, and then we've got fibrinogen and globulin. Most of these are produced by the liver, and they help create that osmotic pressure in the blood. So we said our 3 top plasma proteins. Let's learn a little bit more about each one. Albumins are going to be the most abundant, and they are the ones that truly contribute to the osmotic pressure more than the other 2. And they'll also help to transport molecules in the blood, especially specific hormones. Then we've got our globulins, and they work to transport substances in the blood as well. And another portion of it are gamma globulins, which are going to fight pathogens. And lastly, we've mentioned the fibrinogen, how it's going to become active fibrin, and that helps to form a clot. Now we've got different human blood types, and they're determined by the proteins that are found on the surface of your red blood cell. We are able to perform blood transfusions that transfer blood from one person to the other. And we need to make sure that the blood types, though, are compatible so that we prevent agglutination. Agglutination is when we have clumping of the blood. So I always remember that word glue in here to remind me that it is essentially kind of gluing the red blood cells together and forming clumps. So you could see here we didn't have any reaction. This was a compatible mix of blood, whereas over here we can see that we have agglutination taking place. This person had antibodies that are attaching to the antigens on the red blood cells, clumping them together. So what is an antigen? It's a foreign substance, often a glycoprotein, that stimulates an immune response. I always think of it as an ID for each of your cells. So if you were to have your university ID and you come into the university and you're supposed to be here, that's no problem. It's compatible. But if you have a different university ID and try to come into one of our campus buildings, then you are foreign, right? And we are going to have agglutination take place because you should not be in the building. And so the blood types are determined, like we said, by the presence and or absence of two antigens, and that's type A and type B. So when we have type A blood, we are going to see that we have A antigens as the glycoprotein all over the surface of the red blood cell. When we have type B, we are going to have B antigens all over the cell, and then type AB will have a mix of both, and type O will have neither. So we've already mentioned antigens as that glycoprotein or polysaccharide that serves as an ID. If we see you're a foreign invader, it stimulates an immune response. Antibodies are proteins that are made in response to antigens, especially if they're foreign, and our body is going to then try and latch on to those antigens. Blood transfusion is just the transfer of that blood from one individual into another. And this is just another depiction of the different blood types. Now I know we talked about the antigens that we find on type A blood, type B blood, AB blood, and O, but now let's talk about what type of antibodies each of these people make. If you have type A blood, then you are going to create anti-B antibodies because those antigens are not present on your cell. So your antibodies will not attack your red blood cells. If you have type B blood, you will create anti-A antibodies since you don't have A antigens on your red blood cells. Now, because you have this person has AB blood type and has both A and B antibodies, antigens, then they're going to create no antibodies because if they were to have any of these, then they would attack their own red blood cell. Type O blood doesn't have any antigens, and so their bodies are going to create both anti-A and anti-B antibodies. And this is just in text form of what I described on the previous slide. So during those blood transfusions, if antibodies in the recipient's plasma are going to bind to antigens on the surface of a donated red blood cell, agglutination can occur. So type A can't receive type B blood or AB blood. Type B blood can't receive type A or AB blood, and type O can only receive blood type O. So we think of O as being a universal donor since it has no antibodies on it to attack, or I keep saying antibodies, I'm sorry, since type O blood does not have any antigens on its surface for a different body to attack. And then our universal recipient is type AB since it doesn't create any antibodies. And so it can receive type A blood, type B blood, type O blood, and no one will, I shouldn't say no one, there is no antibodies that will attack it. So we need to ensure that we perform a cross match, which is going to mix small amounts of the blood to test for agglutination before the blood transfusion. And this is just an image depicting that. So we have a type A blood from a donor, and they are going to make anti-B antibodies. And so we will see that there's no binding here. Well, if we have type A blood that's donated and the anti-A antibody of a type B recipient are mixed together, then we're going to see agglutination. And of course, we have to talk about the RH blood groups. So you may have heard of a blood type being either positive or negative. When we have blood type that's positive, that means it has another antigen on here that represents RH. Sometimes this is called antigen D, as in dog. If we don't have this antigen present, then it is going to be RH negative. And we have to think of this in order to prevent hemolytic disease of a newborn. This is when the woman who is pregnant is RH negative, but they have created this fetus with someone who is RH positive, and therefore the fetus is going to be RH positive as well. Now, some of that RH positive blood can leak from the fetus to the mother, and it could cause the mother to create anti-RH antibodies. When the mother later becomes pregnant again, presumably with someone who is Rh positive blood again, and the second fetus is Rh positive as well, the woman's anti-Rh antibodies will then attack the fetus. This can lead to anemia, disabilities, and even death. So to prevent this, the mother should receive a RhoGAM shot, which is going to help neutralize those anti-Rh antibodies within 72 hours of giving birth to an Rh positive child, because through the birthing process there's a mixing of blood. So in that way we can stop these antibodies from developing, so that should the woman become pregnant again, we don't have any antibodies attacking another fetus. And here's just another illustration depicting that disease.